Hello students. Today's topic of discussion is structure of nephron and mechanism of urine formation. Nephron is the structural and functional unit of the kidney, which means every part of the kidney is made up of nephrons, and the function of the kidney is due to the function of the nephron. We know that the kidneys produce urine. It is actually the nephrons which produce urine. Each kidney is made up of approximately 1 million nephrons. Each nephron is made up of three important parts. The first part is the Malpighian capsule. As you can see over here, it consists of a cup-shaped structure which is called the Bowman's capsule, and inside the concavity of the Bowman's capsule there is a knotted mass of capillaries which is called glomerulus. The Bowman's capsule and the glomerulus together form the Malpighian capsule or the Malpighian body. The next part of the nephron is the renal tubule, which consists of the proximal convoluted tubule, which is a small convoluted part or coiled part located closer to the Malpighian capsule. Then the loop of Henle, which is a U-shaped structure, U-shaped tubular structure. This is the loop of Henle. And the last part is the distal convoluted tubule, which is also a small coiled part situated away from the Malpighian capsule. And the last part of the nephron is the collecting duct. So, to sum up, what are the different parts of the nephron? We have the Malpighian capsule, which consists of the Bowman's capsule and the glomerulus. Second part is the renal tubule, which consists of the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. And finally, the last part is the collecting duct. In order to study, before we go to the mechanism of urine formation, let us study about the blood supply to the nephron. You can see that this red color structure marked as afferent arteriole entering into the concavity of the Bowman's capsule. This afferent arteriole is a branch of the renal artery which carries oxygenated blood, but blood that's laden with nitrogenous wastes. So renal artery branches into numerous afferent arterioles. So each such arteriole which enters into the concavity of the Bowman's capsule is called afferent arteriole. Upon entering the concavity of the Bowman's capsule, the afferent arteriole branches to form a network of capillaries called glomerulus. Now, the glomerulus rejoin to form the efferent arteriole, that is the outgoing arteriole, which later branches to form a network of capillaries called peritubular capillaries or secondary capillary network. And the region where it is lying parallel to the loop of Henle, this network of capillaries is called vasa recta. Ultimately, these capillaries join to form the renal vein. Now, coming to the mechanism of urine formation. Urine formation occurs in three steps: ultrafiltration, selective reabsorption, and tubular secretion. Ultrafiltration, which means ultrafiltration refers to filtration under tremendous pressure. It means the blood entering into the glomerulus through the afferent arteriole is at a higher pressure than the blood leaving the glomerulus through the efferent arteriole. And why this happens? Because the diameter of the efferent arteriole is 2 times narrower than the diameter of the afferent arteriole. Because of this difference in diameter, tremendous hydrostatic pressure builds up in the glomerulus. And because of this, the plasma or the liquid part of the blood, along with various organic and inorganic components, get filtered from the blood in the glomerulus to the lumen of the Bowman's capsule. Now, what are the different organic and inorganic substances which get filtered? Organic substances like urea, uric acid, glucose, amino acid, creatine, and inorganic substances like water and mineral ions like sodium ions, chloride ions, and calcium ions. They all get filtered from the glomerulus into the lumen of the Bowman's capsule. This is called ultrafiltration. Filtration under excessive pressure which is built up in the glomerulus lodged inside the concavity of the Bowman's capsule. Ultrafiltration is followed by selective reabsorption. Now the filtrate which comes to the lumen of the Bowman's capsule is called glomerular filtrate or nephric filtrate or primary urine. This is not actual urine. This is referred to as primary urine, but this is not actual urine. So therefore, it is referred to as glomerular filtrate or the nephric filtrate. This filtrate contains certain usable substances like water, like mineral ions, like glucose, and like amino acids. So these substances, they have to be reabsorbed in different parts of the renal tubule. So this process in which these usable substances get reabsorbed in different parts of the renal tubule is called selective reabsorption. So selective reabsorption takes place in different parts of the renal tubule. Like 65 to 80% of the glomerular filtrate gets reabsorbed in the proximal convoluted tubule. Glucose and amino acids are completely reabsorbed, and maximum water gets reabsorbed in the proximal convoluted tubule. What happens in the loop of Henle? The descending limb of the loop of Henle is permeable to water, but it is impermeable to mineral ions. So only water gets absorbed. When we are saying the water gets absorbed, that means the water is moving from the filtrate inside the nephron to the blood. So the water, the descending limb of loop of Henle is permeable to water, so water moves out. Nearly 5% of water is reabsorbed here. Whereas the ascending limb of loop of Henle is permeable only to the mineral ions and impermeable to water. So the sodium ions, the chloride ions, and calcium ions are reabsorbed completely. Now, in the distal convoluted tubule, nearly 13% of the water gets reabsorbed along with mineral ions. So you can see over here different kinds of usable substances like water, like mineral ions, like glucose and amino acids are getting reabsorbed in different parts of the renal tubule. And what's the purpose? Purpose is to maintain the osmotic balance of blood. Or in other words, this particular function or this particular process which is taking place in different parts of the renal tubule helps in maintaining osmoregulation, that is the maintenance of salt and water balance in the body. And that is one of the functions of kidney, apart from playing an important role in excretion, where elimination of nitrogenous wastes are involved. Selective reabsorption is followed by tubular secretion. Tubular secretion is completely an active process, which means it utilizes energy. In this process, substances which are there in the blood, certain substances which are supposed to be removed from the blood because they are toxic to the blood, or maybe they are disrupting the balance of the pH of the blood. So these substances, they need to be removed from the blood to the filtrate, and for that energy has to be spent. So it is carried out by the cells of the ascending limb of the loop of Henle and distal convoluted tubule. So substances like hydrogen ions, potassium ions, ammonium ions, creatinine, and antibiotics like penicillin are removed from the blood to the filtrate in the renal tubule actively. That means by the utilization of energy. And what is the purpose of doing so? This is done to maintain the pH value of blood. So, what did you learn today? You learned that urine formation takes place in 3 steps: ultrafiltration, selective reabsorption, and tubular secretion. Ultrafiltration occurs in the glomerulus. Selective reabsorption occurs in the different parts of the renal tubule, that is in the PCT, in the loop of Henle, and in the DCT. And tubular secretion occurs in the cells of the ascending limb of the loop of Henle and distal convoluted tubule. Thank you.